So the next group of organisms that serve as reservoirs of infections are arthropods. And there's a lot of different types of arthropods that can do this. There's insects, such as mosquitoes and fleas, lice also. And then there's also arachnids, such as mites and ticks, that can serve as carriers of infection. Arthropods are typically referred to as vectors, which means they are a vector of transmission, because they can harbor either a bacterial organism, a virus, or they can harbor a protist organism inside of them. And what the arthropod does is the arthropod will bite a new host organism, such as a human, and when that human is bit, the organism that can cause infection is going to be transmitted into that human. And I'm going to bring this to the front. There we go. So the pathway of, or the chain of infection of the tick, which what we outlined earlier in terms of transmission of the infection known as Lyme disease, or the disease known as Lyme disease, occurs because of this Borrelia burgdorferi bacteria that resides in the gut of these ticks. And so once these ticks are infected with the bacterial organism, they can pass it through their saliva into a newly infected host after biting them. So that's how Lyme disease is spread. Malaria is another example, which is spread through mosquitoes. And what a mosquito will do is it will take a blood meal from an animal, and then it will go out and take another blood meal from a human. And when it does so, it will pass on these protist larvae that are called plasmodia. And the plasmodia will start to develop inside of the human host and start attacking liver cells. And the plasmodia will start to replicate and eventually leave that human when another mosquito bites it, causing the development of malaria inside the next infected individual. So those are just some examples. And here's a distribution map for areas that are at high risk of where an individual can contract Lyme disease. The colors on here aren't all that great, but if you look at the scale, the darker pinker regions, according to the map, are going to be moderate risk, but the black areas is where it's a very high risk of contracting Lyme disease, which is great for us because we have a very low minimal risk of contracting Lyme disease in South Carolina. So that's a distribution map of Lyme disease. Another source of infection are going to be that of fomites. Fomites are inanimate objects that are have the capacity to transmit pathogens, and these can be things that we just come into contact with. It could be your pencil, it could be your notebook, it could be surgical instruments, it could be touching the hand sanitizer dispenser inside of a clinical setting. An inanimate object that has pathogens on it that can be transmitted is pretty much all over. And so these essentially are going to be anything that's non-living. So a non-living reservoir, remember, is the other type of reservoir aside from the living reservoirs that we've discussed. This also includes air, soil, dust, water, and foods. So all those are considered non-living. And then the biggest thing are going to be fomites, which again are inanimate objects that have the ability to transmit pathogens. Bed linens in hospitals are a great example of fomites that can harbor a lot of diseases with them. Blankets, pillowcases, really anything that a patient has come into contact with that could have easily been laden in those bedding materials, which is why we have to make sure we decon those materials and not reuse them in a clinical setting. One example of kind of organism that can be transmitted through water, because we're talking about non-living reservoirs, is that of Giardia lambda. Maybe you've heard of this before, but it's an organism that is passed through inanimate objects, or it's passed through a non-living reservoir, primarily water for many people, but it can also be passed through food that has been contaminated. And so what happens with this particular organism is that the organism begins its life stage as a cyst and develops quickly into this new kind of form called the trophozoite. And the trophozoite can easily infect the patient that it comes into contact with. So what first happens is you have the generation of a cyst, and the cyst is going to colonize either an apple, berries, or any type of food, pears. That's what we see here. But most people pick up this organism from drinking contaminated water. So a contaminated water source that hasn't been filtrated properly, and proper chemicals haven't been used to treat the water. So you have the cyst Giardia inside of this water, and then a human consumes the water. So say you're hiking in the woods and you run low on water, and then you pass this nice free-flowing stream. So you bend down and you pick up a sample of the water and you put it in your mouth. Well, you may ingest some of the cyst, and in doing so, the cyst is going to make its way through the digestive system, and as it does, it's going to generate in the form of this trophozoite. The trophozoite is going to mature, and it's going to proliferate, and it's going to divide, and when it does this, then it's going to elicit infection and cause a lot of gastrointestinal complications for the human that's now being infected. So Giardia infection is no picnic. The individual that suffers from this is going to reflect a lot of gastrointestinal complications or gastrointestinal distress, such as the releasing of diarrhea, the vomiting, and a lot of nausea in the abdominal region. And so this again is an example of contracting an organism through a non-living reservoir, and in this reservoir this is going to be essentially water, or it could be food. Now the next inanimate object that we can kind of look at an example for infectious transmission is going to be that of fomites, and we just have on this image a lot of different instruments that can serve as fomites. So eating utensils, surgical equipment, as I said earlier, needles and syringes are big things, stethoscopes, bedding, wound dressings, cups, blood transfusion equipment. So all of these in a clinical setting can definitely serve as reservoirs of infection, which is why decon and hand hygiene are pretty important aspects that are required to help us to control infections or infectious agent outbreaks. Let's talk about the most common mechanisms of transmission, and those are going to be things like direct skin-to-skin contact, so directly touching or coming into contact with individuals that are infected with an infectious agent. Mucous membrane to mucous membrane contact, so this can be pathogens that are transmitted through kissing or through sexual intercourse, but that mucous to membrane interaction is a very easy method of transmission for infectious agents. Some other common modes of transmission include indirectly contacting airborne droplets or respiratory secretions. These are things that are put in the air through the process of sneezing or coughing. Indirectly coming into contact with food and water, as that has been contaminated with fecal material. So the things we eat, in which we're very cautious about what we eat, we really have no idea as to where they've come from. But if they haven't been sanitized properly, we can put ourselves at risk to indirectly come into contact with infectious agent. Arthropod vectors, which we've talked about already, fomites we've talked about, and then lastly, there's also indirect contact via transfusion of contaminated blood or blood products, or using syringes and needles that haven't been sterilized properly. So those types of mechanisms can be employed in order for infectious diseases to be easily transmitted, which is why we make every effort and take precautions to prevent this from taking place. Here's a diagram kind of highlighting HCV infection. So HCV infection, also known as hepatitis C infection, is an example of, we're going to use this as an example to talk about transmission. And what this is showing you here is kind of how HCV infection is acquired in a time frame before the 1990s, and then how it was required in a time frame after 1995. So if you look at the majority of this pie chart here, you can see that drug use through injection was a primary method that enforced the transmission of hepatitis C in the population. So it took up made up 60% of how the virus was transmitted. Sexual contact represented 15%. Blood transfusions represent 10%, and then we have some unknown other, and then occupational, which would be something like a needle stick inside of a laboratory setting. So those are different specific modes of transmission, but what you can see here is that injection through drug use was the key culprit in terms of transmitting hepatitis C among the population. Hepatitis C infections acquired after 1995, again, we see an increase in injections-based infections at 68%. It looks like here we've got an increase in sexual activity to 18% in terms of transmission. Occupational, other, and unknown are still relatively the same. So we do see an increase here. The other categories are going to be things like mother-to-infant infections, or you can have infections that are related to medical care acquired, such as nosocomial infections. So HCV infection, we're just kind of looking at this kind of as an example of the different common modes of transmission that are out there. So again, that's transmission through blood transfusions, occupational encounters, unknown sources, and then primarily for this particular virus, we're looking at injection through drug use. So let's go back to looking at agencies that are in charge of monitoring infectious agents in the population. I'm going to start off talking with the World Health Organization. World Health Organization, which is a part of the United Nations that was founded in 1948, and their job is to kind of create a platform by which all nations are able to cooperate and come to a decision on how to deal with outbreaks, epidemics, pandemics that are occurring in areas, and to generate policies and programs that can hopefully control these disease outbreaks, to either eradicate the diseases or control them, so that way members of the population can better survive. And this obviously is coincides with the ideals of the United Nations, which is to improve the quality of human life. So example investigations that WHO has been a part of include outbreaks of Ebola virus, the eradication of smallpox, which is the only virus to have been eradicated to this day and time. And right now the big key thing that the WHO is working towards is eradication of polio, as well as draculiasis. which is also known as guinea worm infection.